Okay, so let me introduce Sadik. Dr. Sadik completed his PhD. Dr. Mohammad Sadik completed his bachelor's and master's degree in biochemistry at Aligarh Muslim University, Aligarh, India, and received his doctoral degree in molecular and structural biology at Central Drug Research Institute, also called CSIR-CDR in Lucknow, India. His PhD thesis focused on characterizing the biogenesis of mitochondrial iron-sulfur clusters in Plasmodium falciparum. In 2021, he joined University of Maryland, Baltimore Department of Microbiology and Immunology as a postdoc fellow. His postdoc work focused on elucidating microbial pathogenesis, protein secretion mechanisms, and host-pathogen interaction. A significant achievement of his work is the identification and functional characterization of secretory lipases that contribute to rickettsial infection processes. Currently, his research investigates the role of outer surface and secreted effector proteins with particular emphasis on their interaction with the host components for adherence, internalization, and internal growth. Another project he's currently working on is investigating the role of transfection factor EB, also called TFEB, in regulating lysosomal biology during rickettsia infection.
Thank you so much, Dr. Abid, for this wonderful introduction. So I'll start my presentation. So today I'm going to present my work, what we are doing here in UMB. So actually, basically, we are working in the Department of Microbiology and Immunology here in UMB, and our work focused on the rickettsial pathogenesis. That the rickettsia is a bacteria, intracellular pathogenic bacteria that infect the host cells, endothelial cells, and how it's get internalized and colonized into the host cells. That's the primary focus of my work. So, as rickettsia is a infection, obligate intracellular bacteria that naturally maintain into different arthropods like tick, flea, and mice, and these cause of vector-borne diseases, and they account for more than 70% of all the infectious disease. And there's rickettsia alone, rickettsiosis, the disease caused by the rickettsia is called rickettsiosis, and the rickettsia alone cause for the 25% of all cases in the United States. Here I have listed some of the diseases called vector-borne diseases. Our focus is to the rickettsiosis. So the poor understanding of this intracellular lifestyle of rickettsia, as well as the unavailability of vaccine, the effective vaccine against this rickettsiosis, making the situation even more worse. So, as I told you, rickettsia is a zoonotic pathogen, and on the basis of species-specific pathogenicity and disease development in human, it can be categorized into highly pathogenic, and non-pathogenic rickettsia. Here in this figure, we have organized all the different rickettsial species on their pathogenicity, with the highly pathogenic on the left side and the non-pathogenic on the right side. So, the intriguing question is how some of the rickettsia are pathogenic while other are not. There is little information is available about all those things. So the one answer might be like bacterium-derived effectors. So in our study, I will use this one highly pathogenic Rickettsia rickettsii and one pathogenic Rickettsia parkeri and one non-pathogenic Rickettsia montanensis in our study. So this is a general model for host rickettsial infection to the host cells that involve starting from the attachment, followed by internalization, and then evasion of lysosomal fusion to establish a cytosolic replication niche. This is a very complex process, and to regulate such a complex process, rickettsia use an arsenal of effectors. And through our study and from our lab and research from other lab have been identified a lot of effectors like RISC1, that is a kinase, PET1 and PET2, that is lipase, sorry. And then there is another RELF. All these effectors help the rickettsia to get internalized and establish a cytosolic replicative niche into the host cells. But these effectors. Some of effectors are present in some rickettsial species and some are not. So, and some of them are like dispensable also. So, because of their sporadic presence as well as the dispensability, we hypothesize that rickettsia may possess some additional membranolytic enzyme that help the rickettsia to internalize and make a replicative niche into the host cells. So, to test the hypothesis, we search on rickettsial genome using web-based server FireA2, and we identified a secreted lipase with hydrolase motif, and we named this as rickettsial lipase RLIP. And we did the sequence alignment of RLIP with previously characterized lipases and phospholipases, and we found that this serine hydrolase motif, we call it GXSXG. This serine hydrolase motif is conserved among all the previously characterized lipases. And we also did this RLIP sequence alignment of RLIP from different rickettsial species, and we found that this serine hydrolase motif is almost conserved among different rickettsial species. And as lipases belong to alpha beta hydrolase family, we make a homology model of this RLIP by some using bioinformatic tool, and we found that RLIP shows a classical lipase structure with a beta sheets surrounded by two alpha helices, and their active site GS active site, like all hydrolase motif, is present into the loop between alpha helix and beta sheets. And here I have highlighted this active site serine S138A, as I'll use this active site serine will do the some mutation analysis also for this active site serine. So on the basis of all these bioinformatic analysis, we hypothesize that RLIP is a putative lipase with a conserved serine hydrolase motif. For the functional characterization, we recombinantly express and purify that RLIP wild type as well as the active site mutant protein, and we confirm the identity of protein by Western blot as well as the that sequence analysis also. And we check their lipase activity using lipase assay kit, and we found that wild type lipase has a lipase shows lipase activity. However, the mutation of active site serine at 138 position into alanine greatly significantly decrease the lipase activity. We use LAGZ and positive control as a reference. So on the basis of these findings, we suggest that RLIP is a functional lipase, and serine residue of actives of their hydrolase motif is important for their lipase activity. And given that phospholipases plays an important role during host cell infection, we proceed to determine the transcriptional pattern of RLIP by RT-PCR. We use two cell line, Vera and HMAC. Vera cell line is most commonly used epithelial cell line for the maintenance and doing some study of RLIP rickettsial infection, and HMAC represent the primary target. HMAC is the primary target cell line for the rickettsia. So through RT-PCR, here we here we show that expression of RLIP in a black line and the bacterial burden in red line. And we found that in both of cell line, the RLIP expression increased during early stage of time, during early hour like 15 minutes, 30 minutes, and remain elevated during the course of time. And bacterial replication increase after like doubling time, like after 24 hours. So our data demonstrate that RLIP transcription increase during early stages of infection and is remain elevated during the course of infection. And then we check whether this RLIP is secreted to the host cells or not with like cellular fractionation assay. And we found that RLIP in both of cell line, Vera and HMX cell lines, RLIP is secreted mostly to the cytosol with minimally retained by the bacteria itself. We use PAT1 as a positive control and use OMP as a rickettsial surface protein that suggests that and the presence of OMP only in the pellet fraction of infected cells suggests that our that fractionation approaches didn't lyse the bacteria. So due to this intriguing expression and pattern, we proceed to check whether early protein expression induced during rickettsial infection or not. So we infect that vera cells or host cells with with the rickettsia and collected the sample at different time point and check the protein expression by Western blot. And we found that purified rickettsia itself has minimum level of protein expression in it when it is outside the host cells. But as soon as it is attached to the cells, its protein expression get increased. So on the basis of these uh, uh, data, we suggested that early expression was early was minimally expressed inside the bacteria when the bacteria is outside the cell. But as soon as the bacteria infect the cells, their early expression start increasing. So, we start to evaluate the protein expression pattern of RLIP during the host cell infection using different pathogenic bacteria. Like we use two pathogenic Rickettsia and Rickettsia parkeri and one non-pathogenic Rickettsia montanensis. And we found that the RLIP expression was higher in highly pathogenic bacteria. However, in montanensis, their expression is very low. So we suggested that this protein is highly expressed only in the pathogenic bacteria, but not in the pathogenic, non-pathogenic. So we check whether this protein expression difference in their protein expression is because of the bacterial burden or protein expression level. We confirm the protein expression as well as the bacterial burden using RT-PCR, and we found that the RDP expression was very low in non-pathogenic rickettsia, and it's remained low and unchanged. However, in the pathogenic rickettsia, the RDP expression was high, and the bacterial burden was quite almost same in all three bacteria, except other than like in for Montanensis, it is little bit low, significantly low, but yes, it is low. So on the basis of this uh, data, we suggested that RLIP is differentially expressed among pathogenic and non-pathogenic rickettsia and may contribute to the infection process of pathogenic rickettsial species. Now we proceed to check whether this RNIP expression, if we get remove the bacterial the early from bacteria, what we'll get. So the the to study the gene specific function by using traditional genetic manipulation approaches were not easily available for intracellular bacteria. So we collaborated with Dr. Erin Goli in from John Hopkins. She provided us a bacteria, a mutant bacteria where RLIP was not there. So we confirmed that mutant rickettsia by RT-PCR as well as Western blots. Like we can see that this is early protein. It is present in wild type, but in mutant bacteria there was no, like there is a truncated protein and no functional lipase was there. And instead of lipase, we got a truncated version of protein in mutant bacteria. So we determine the protein expression, followed by we monitor the expression pattern of RLIP in wild type as well as mutant bacteria. And we found that in wild type bacteria, there is a significant increase in RLIP. However, in mutant rickettsia, there was a very low or unchanged expression of RLIP. And then we check the bacterial burden, and we found that bacteria, RLIP wild type bacteria grow well in host cells. However, the mutant bacteria didn't grow well. So we check whether this observed difference in the bacterial burden is because of the internalization or the following process like escape from vacuolar. We did the differential staining as well as staining with LAMP2 marker. And we found that during differential staining, we didn't find any significant difference between wild type and mutant bacteria. So this data shows that the RLIP didn't affect the internalization process of rickettsia. However, when we see the staining with the LAMP2, we found that most of the most of the mutant bacteria is localized with the LAMP2, suggesting that all the mutant bacteria is remain enclosed to the phagosome, and it will be removed eventually by the fusion with lysosomal fusion. So on the basis of this finding, we suggest that RLIP doesn't seem to help the internalization of bacteria. However, it shows that after internalization, it avoid the clearing of the bacteria by lysosomal fusion. So, on the basis of all these data, we suggested that RLIP is a functional lipase and that secreted to the host cytosol and expressed during early stage of infection, and that help the bacteria to grow by avoiding phagolysosomal destruction. I acknowledge my lab member, like my PIs, supervisor, Saidur Rahman and Oliver Voss, and then all the collaborator, and the research is funding by these funding agencies. And I'm happy to take questions.
Thank you, Sadik, for an exciting talk.
Thank you so much.
We'll wait for the questions if anyone has any.
I have a question.
Yep.
You are talking about TFAB, no?
Yeah. We started about
Yeah.
we started we started that TFAB project. Like we got a HeLa cells mutant with the TFAB, and we did some initial studies, and we checked like if we grow the cells in normal HeLa cells and in the TFAB knockout, we found that in TFAB knockout the cells the bacteria grow well as compared to the wild type HeLa cells. Yeah, so we are working. We just started those TFAB experiment. Yeah, so yeah, go ahead with the question.
Does it have to do anything with the translocation of TFAB from cytoplasm to nucleus?
Yep, exactly. Yeah, so I think couple of week before I did the experiment of translocation. So, and we found that we found that after rickettsial infection, the TFAB is not that much localized to the nuclear fraction. It mostly to the cytosol, because if like what we assume from that data that if rickettsia activate TFAB too much, if too much activation happen there, so TFAB will go to the nucleus and it start making that lysosome, and then lysosome comes to attack on the rickettsia, and then rickettsia eventually die. So what we hypothesize that is might be not enhancing too much that TFAB activation for their own survival. Yeah. We just started those projects. Yeah.
Yeah. Does it have to do anything with the autophagy as well? Because both are related, no?
Yeah, both are related with the autophagy. I don't think we didn't do anything yet. Maybe we are thinking that it doesn't help starting the autophagy, because if we start induce autophagy, that will be like a negative thing for the bacteria. So I think maybe we'll see soon. Yeah, because we didn't do too much.
Question may arise, because both processes are interrelated, no? So
yeah,
it's
both
definitely having some effect on that one. So reviewer may ask, so I think it's better to check.
Yeah, we'll go ahead. I just did two, like I did RT-PCR and Western blot and cellular fractionation for TFAB. Like last month we started this TFAB whole project. Yeah. Thank you so much for that.